I'm Carika Weldon from the University of Oxford as a scientific researcher, and I'm here today with Karla Neugebauer from Yale, and she's going to tell us all about her fascinating research. She had your talk yesterday, and you had it today, this morning.
It seems like yesterday.
I know, it's been such a long day. It's been a great day of science.
Fabulous.
Can you just give us a nice snippet of what you talked about this morning?
Well, I talked about some new data, and I'm really excited about the new approach that we're taking. We're sequencing Nascent Rna, which is like the very first RNA. As it starts to get synthesized by RNA polymerase II, we purify it and we sequence the whole length of the RNA, and that gives us insight into that whole life history of each individual RNA, because we can see where transcription started from the 5' end of the sequence, and then we can see where the polymerase is sitting when we isolate it from the 3' end, and we can see all the things that might have happened to that RNA in between, like splicing, and splicing is one of my strongest interests. So
me too.
We're really excited about being able to see the dynamics of nascent RNA, because it's just this amazing species of RNA that has all these different 3' ends and all of these different sequence features due to the changeability of the RNA in the body, in the body of the RNA. So we really feel like this is the tip of the iceberg, basically, that we would be able to associate the progress of transcription with the progress of splicing. But then finally, there's going to be a whole avenue of interesting research that'll be about folding or RNA modifications or changing RNA binding sites as exons get ligated together. I just feel really sure that this is going to be a really important regulatory kind of gold mine where we can see what's actually happening to nascent RNA.
I was really excited with your talk because you mentioned long read sequencing.
Yeah.
And it's something that, is kind of picking up steam over the last few years. And, and then the other part of it is that when you learn about, gene regulation, you learn about transcription, then you learn about splicing, and then you learn about translation, and you kind of see them as these three separate events. But then to see that actually, it's all happening together, and you have some really beautiful images, and it was just really nice to see that it's not these three separate events, but that you have such great data. Can you talk more about that, about your actual research that shows this is all happening all at once?
Well, I think the wonderful thing about being a biologist is that you go to do an experiment and you think of, maybe 1, 2, or 3 hypotheses, options on what the data is going to look like. And that's why you're producing the data. And then you produce that data and you look at it like, initially, and you're looking for those 3 things, and sometimes, usually you're completely blind to that other thing that you're actually staring at, and you just don't even see it. And then some time later, you go, oh my God, what is that? And then you realize that you're seeing something unexpected, and of course, that's usually the most interesting thing.
Exactly.
So the thing that we discovered in the most recent study that we published was a study in SPAMI. And that was one where we expected to be able to ask, are individual introns in the same gene removed in their order of their synthesis, or is it all mixed up due to regulation? And what we found, we looked at that, and it turned out that it can be either in order or mixed up. And there was evidence for kind of both modes. But what was the surprise was the presence of transcripts that were completely unspliced, and then the polymerase read through the poly A cleavage site and just kind of kept going. And we had obviously no reason to suspect that there would be this population. But of course, if you were doing short read sequencing on nascent RNA, you would never know that because the retained introns would simply be added to all the numbers that you're tallying up for the splicing frequency of each individual intron. You'd never realize that they were all in the same transcript. So that I think that really illustrates the power of the long read sequencing, and then your ability to link, for example, all those splicing events with each other, but then also with poly A cleavage, the fact that the polymerase just never stops. So I just really love this method, and so now in our new study, we're also seeing things that we didn't really anticipate. So I just think it's a wonderful voyage. into RNA.
Yes,
my favorite molecule.
Me too. So one of the things that really got the crowd going this morning was you had a slide and you said, I need you to use your imagination because I can never get an animation that seems to capture mine.
Right.
Do you think that day is going to come when you're going to be able to capture what's happening in your mind? They're going to have the full story and you can have a great slide to convey that.
Well, so exactly. So I have sat down with pen and paper and with Illustrator and with PowerPoint and tried to illustrate these things. But, you know, one thing, I love illustration, but on the other hand, I feel like when I draw it, it's a little bit too concrete because the thing that's captivating to me is the notion that there's this whole continuum of possibilities.
Right.
So I can't actually illustrate all of the different possibilities. But if I ask the audience, I finally decided, I was wrestling with this yesterday, like, what am I going to do in my talk? Like, I really want people to try to imagine all of these dynamics happening at the same time. The fact that when two exons ligate together, they create a new sequence at the junction. They've lost this big intron, so now that sequence is gone.
Exactly.
You know, that's like exon 1 is now completely different. Exon 1 is a totally different ball of wax. And so I don't know how to illustrate that without it making it be too concrete also, because I don't know what that sequence is, you know, like I don't want to do that. So I also think from a public speaking point of view, you know, if you can really, it's kind of common ground with the audience, because I'm sure everyone in the audience has had those same experiences where it's hard to communicate an idea by drawing it.
Exactly.
So if you just like look the audience in the eye and go, you know what, I just don't even know how to diagram this idea. So can you
help yourself? Like a buffet?
Right, exactly. So I don't know, it seems to work.
I think it does. And I think
it's a comic relief.
It is. And I find, you know, on that topic, diverging a little bit, but as a scientist, you have public engagement, this word that's kind of thrown around a lot, and you find that there's some people that are really good speakers as scientists, including yourself, but then there's some that are not as great, right?
Right.
Do you think that impacts on how science has kind of developed? Because if other people are not able to capture the imagination, as you put it.
I think that it's a definite plus to be a charismatic speaker, to be able to communicate your science. I don't know if you know what I'm talking about, but sometimes there's a speaker who you'll see repeatedly at meetings who's not very good, not a very charismatic speaker. You know, kind of mumbles and swallows their words, and you're just thinking, like, why is this person at every meeting I go to? But it's because their science is so cool.
Right,
so they,
exactly.
you know, clearly scientists are, I don't want to say we're a bunch of nerds, but we also have a culture where it's not just about selling an idea and being slick. It's about having good ideas and doing a really beautiful job. So the person who I was actually thinking about and won't name is a wonderful scientist, very imaginative, and just has amazing data. And so even though he's not a charismatic speaker, you know, it would be great if he were also, but everybody loves to listen to him and experience the science that he's been, you know, promoting in his lab. So so that's one of the things I really like about the scientific community is that it's very inclusive of a lot of different personality types, even though clearly somebody who's like really confident and kind of pushy, you know, can get maybe more attention than the next person who's more retiring. I do think that the scientific community is really mainly about the science and interested in the science, and I don't know, I just hope that mostly adheres to that.
Yeah, and kind of piggybacking off of that, obviously, I mean, this is the 84th Symposium.
Amazing.
84 years ago, I definitely wasn't around.
No.
I don't think you were either.
No.
And I mean, back in that time, you know, there was a big difference when it came to women in science, right? And so we've come a very far way with that as well. What do you think about that?
Well, it's funny because I worked in Europe for 13 years and I was involved in promoting women in science in Europe where I guess a comment about Europe is that certainly in Germany where I was working, and Germany is a wonderful environment for doing science, but I think it's hard for women especially because there's no sort of career ladder where the expectations for each of the steps,
right.
And where you don't necessarily have to move cities in order to get to the next rung on the ladder.
Right. Yeah.
I think moving is very stimulatory and it's a wonderful to be able to move, but I think that it's frightening for some people who would be excluded, right?
Right.
If they just, why should your willingness to move your family, in other
right,
words,
yeah, this is true.
Be a criterion for allowing someone to be a leader in science? That doesn't seem like a rational choice. So I feel like that in Europe the career structure isn't very helpful, and you can see that because there are really women missing from the leadership. So that said, coming back to the U.S., and I think women are better represented in the U.S., I now can see with more open eyes because I had been dealing with that and trying to increase the visibility of women in Europe. I started a database of expert women scientists.
Oh, that's perfect, yeah.
Which was meant to actually, kind of ironically, was meant to show off women who'd actually been achievers and achieved a lot, to say, look at these, you can't tell me you can't invite someone to give a seminar, because what about one of these 800 women in the database,
right.
So that was the idea of it. And actually Kai Simons, who was the founding director of the institute that I worked at, he asked me to put up this database, and so I did that. But so coming back to the US, I sort of thought, well, there are more women in leadership positions in the US, so now I'm scot-free. I don't have to deal with this issue anymore. And now I kind of realize, that we're going to be dealing with this diversity issue for a really long time. Because first of all, women aren't equally represented in science sort of as you progress through the career track. And there are other diversity issues as well with underrepresented minorities and so on, where clearly we want to make sure that every person who wants to be a scientist doesn't feel that they don't belong. So I think that's a main goal that I have, and I try to express that also at Yale when I teach a lot of undergraduates. I'll have actions in my classroom.
Ooh.
And an action in my classroom at one stage.
What is that?
Actually, it was really funny because the students spoke with me about, I think there was a sense of cliquishness in the classroom that some of the students who were not in the major, but were outside of the major, but it clearly, like from the chemistry major or, absolutely capable and credentialed.
Of course, yeah.
Just to give an example, there were a number of different students who felt like they weren't in the kind of mainstream of the classroom and that they were at a disadvantage. And so I spent 15 minutes at the beginning of the next class. I showed the Joan Steitz's Lasker video.
Okay, awesome.
Where she talks about identity and her identity as a scientist.
Yes.
That, that's who I am. I could define myself in any number of ways, but that's the bottom line for me, that I'm a scientist. And I just find that statement that she makes so moving. And so I played that for the class, and I asked them about their identities too, like who's an athlete.
Okay.
It turns out that athletes in college think that they're dumb.
That's the stereotype, yeah.
Because they think I was recruited because I can, catch a football or what have you, or hit a tennis ball, but that is so not true.
It's not, yeah.
They wouldn't get accepted to Yale University if they weren't capable of doing the work. It's absolutely, I was sort of gobsmacked when I realized this. So anyway, so that was my action. And some of the students expressed their appreciation for doing that. I just really feel that sometimes we in the audience, and that can include me at a scientific meeting, sometimes I really feel like, oh my God, I don't really belong here.
Yes.
And I think it's important to recognize also that we can kind of work ourselves into a corner where we think that we don't belong, and when we do have a right to be here. We do have the credentials.
Of course.
we could like face that.
I know when I went to my first meeting, right? This is my first Cold Spring Harbor meeting, by the way.
Oh, it is?
It is.
Oh, wow.
And I'm like, it's so much. But it's really a good so much, right? But I remember when I went to my first meeting, I was just like you said, how did I end up here? What am I doing here? But you get to realize, speaking to people, and I'm sure you might be able to relate, that even though you have a professor, he's published all these papers, he's still eager to go around to all the posters, speak to the graduate students, because he's not an expert in everything. No one knows everything.
Right.
And that's the joy of being a scientist. No matter what race, sex, whatever you are, as a scientist, I think the main thing I realized was when I got my PhD, it just proved that I don't even know what's going on. Like, there's so much stuff I don't know.
That's why it's a doctor of philosophy, because you have to be philosophical.
You have to realize that, you know what, the more I delved into this one topic, the more I realized I know nothing about everything else, or even about my topic. It's like you realize that you don't know anything.
Well, that's the fun of it, right?
Yeah, awesome. So as a scientist, where your work is now, you're really excited about the long read sequencing and where that could potentially go. Where do you see your field in about 15 years?
15 years?
Yeah, so let's say at the
oh my God.
At the 100th Symposium, what would you like to see?
Oh my God. I mean, I guess I'm going to do an exercise where I look back 15 years ago and I ask myself, like, what would I be shocked at, like now, if I were, just the fact that a splicing disease like spinal muscular atrophy can be cured using an antisense morpholino, Adrian Krainer's work. I could not possibly have imagined that back then. Maybe he could have.
Maybe.
It's pretty amazing. And also the thing that I try to convey to my students that's really fascinating, when Joan Steitz discovered the SNRPs, there were these small RNAs that assembled little particles, and they went off and they did splicing. Look at how many other small non-coding RNAs are in RNPs, where the RNA does the business.
Right.
CRISPR, telomerase, SNO RNPs, microRNAs in the RISC complex.
siRNAs.
PWE, siRNAs, exactly. So, that's just amazing. That's really amazing. I mean, so there could be more of these things. At the moment, I feel like, oh my God, they must be all known. But it could be.
Exactly.
I guess it could be. I think that the one thing that I feel is a real black box where I hope we're going to go is where their dynamics, like, an example that's close to me, but could be something else, where there's, a progression of events that must happen to this thing, like splicing, and then you make an mRNA, but where you actually don't, there are black boxes around, what happens if it doesn't do this during this time period? Does it happen later? Does the thing degrade? So I think that we definitely need to develop new methods that make our understanding of populations of molecules and the sort of quantitative aspect more accessible, so that we can also interact with physicists and mathematicians and get more quantitative and predictive about RNA biology, because that's something that's going on in things like mechanical biology and cell motility and so on. And I think in RNA biology, it's not going on quite as much.
So my last question would be, if you could say something to inspire the next generation, what would it be?
Oh my God.
So if I reword it, if you take yourself back, can you remember what inspired you to follow this track, to be a scientist?
Well, I think that it was just actually what you described, is the sense of wonder as you discover something, and then that sort of vanishing period of time when you feel you sort of feeling kind of chuffed that you've discovered something, and then you see that there's just all these additional questions.
Yes.
And it's that problem solving that's so much fun. So it's just like, you know, a bouquet of flowers blooming.
Bouquet of problems.
Right? And so I think that's what all scientists must find so enjoyable, and especially as a young scientist to experience that, let's say for the first time or for several times, is it was always amazing, but I think for young scientists must just be magnificent, and so I hope young scientists continue to do PhDs and postdoctoral fellowships, because with science training you can go into so many different directions, but
Exactly.
then to actually have the opportunity to discover some things is it's a wonderful life experience.
Thank you so much. It's been a pleasure talking to you today.
It's been great talking to you.
Thank you.